it's a distinct pleasure and a privilege to introduce Dr. Mark Oman, who's going to talk to us today about one of the most amazing phenomena on the surface of the planet, the migration of zooplankton in the ocean's interior. Mark is a professor here of biological oceanography, and he's also the curator of the SIO Pelagic Invertebrate Collections, and you're going to learn more about pelagic invertebrates in just a moment. Mark is a very active researcher in the field of biological oceanography with programs here on the West Coast studying the California current using a long-term data set, and in the Northwest Atlantic and the Norwegian Sea. So without further ado, Mark, thank you very much for joining us.
Okay, is the mic on? Yes. Well, I'd like to thank the Birch Aquarium for the invitation to speak here about a truly intriguing phenomenon, and thank all of you for coming at this bright hour. But as you'll find, the day has long since dawned for the animals that we'll be talking about today, the oceanic zooplankton. Now, I'd like to set the stage for my presentation by thinking about migrations in general, animal migrations in general. And when we think of migrations, we tend to think of the large-scale horizontal displacements of animals across the surface of the Earth. We think of migrations such as the monarch butterfly moving north-south, perhaps Canadian geese and their annual migrations, the California gray whale migrating from the Bering Sea feeding grounds to the calving grounds off Baja California, these large-scale geographic displacements on the surface of the Earth. Well, I'm here to tell you that I think the most remarkable migrations are actually vertical migrations, not lateral ones, and they go on in the ocean on a daily basis. Now, one of the phenomenon has actually been known for well over a century, but one of the early tools that was used in the study of animal migrations was sonar. And what I'd like to do is briefly roll the clock back 60 years. The year is 1942. There's a war on. There's considerable interest in the physics of sound propagation in the ocean. This is of obvious importance for echolocation, bottom finding, recognition of objects of various kinds. And a great deal of research was conducted here at Scripps concerning acoustic wave propagation in the sea. Some of this was done right offshore in these nearshore waters in an area of the San Diego, called the San Diego Trough as well. Now, this is a sort of schematic of what the sonars do, is to send a pulse of sound from a ship at the surface downward into the ocean. The sound travels quickly through seawater, reflects off the bottom or other surfaces in the water, returns up to the ship. Now, at the time that this work was being done, about 60 years ago, some unusual phenomena were uncovered that surprised the physicists who were trying to understand acoustic wave propagation. And this is an echogram of about that time period, a little bit later, but close to that time period, and something resembling what they might have seen in that era, where here's the sea surface right here, and here is a sharp acoustic reflection off the sea floor. Okay, that's very well defined. But mid-water, there's something rather peculiar. There's a rather diffuse layer that appears with, it's unlike the characteristics of the sea bottom, which are, in this case, was a very well-defined sharp reflection. This instead is rather a more scattered sort of appearance. It's a diffuse scattering layer, and hence the name deep scattering layers. So sound is reflected here, but it's scattered here. And there was great concern about what the cause of these deep scattering layers might be. It was thought that there were probably physical phenomena that might account for it, so a temperature or density discontinuity in the water column. But there were complications because these layers tended to be more visible, more apparent in the daytime, and tended to be less recognizable at night. And there were various hypotheses put forth about what might be causing this daytime aggregation of these layers. And by analogy with the ionosphere and the atmosphere, people thought there were physical phenomena that were causing sound to propagate differently day and night. Well, it took a biologist to solve the riddle of the deep scattering layers. And biologists have solved the most interesting problems in the sea. And this was a professor at Scripps named Martin Johnson, also one of the authors of the celebrated book, The Oceans, who quickly realized that this kind of pattern of daytime formation, and now I've introduced sunset here, nocturnal ascent just about the time of sunset up toward the surface, and then we have a little gap in the record. The layer is up near still up near the surface, and then just before sunrise, the layer begins descending again. That this phenomenon had a biological origin, and it's attributable to a variety of different kinds of organisms. In the case of the frequency of sound that was being used at that time, it would have been mainly these shrimps, euphalsid shrimps, surgestids, myctophid lanternfishes, squid, and a variety of other larger organisms. Okay, so understanding the biological source of these deep scattering layers was a very important discovery. But what that doesn't address is the question why animals would migrate vertically, why they would move up and down in these layers, reproducibly coming up near the surface and going down into the interior of the ocean, every day, every night. Why migrate vertically? Why go up and down rather than horizontally, or simply remain at a fixed depth? And I'll try to help you understand why that might be this morning. Now, the animals that I'll be talking about are not the actively swimming organisms, the fish and squid and so on that we call nekton. Rather, I'll be speaking about the plankton, the zooplankton in the sea. Plankton comes from the word planktos, the Greek planktos, meaning drifter or wanderer. And these are organisms that are largely at the mercy of ocean currents. They have swimming ability, but not sufficient swimming ability to swim against ocean currents. And to convince you that that's true, I'm illustrating a krill, Euphausia pacifica. The geographic distribution of Euphausia pacifica, this is Japan here, the Aleutians over here, British Columbia, and we're right down here. And you see that the large-scale distribution of this species of zooplankton is confined to one geographic area, one broad geographic area. And this, in fact, corresponds to the major surface currents of the ocean. The west wind drift moves across here, enters the Gulf of Alaska, part of it enters down into the California current. So the ocean currents, to a first order, dictate the distribution of these animals. However, I want to convince you that despite the fact that they're moved horizontally by ocean currents, they do have vertical swimming ability. And this will be just a brief video clip taken from the research submersible Alvin, right out here in the San Diego Trough, by colleagues of mine. As the submersible ascended back up toward the surface, having done some experiments on the sea floor, they turned the lights on and turned on a video camera for me and recorded the zooplankton and other particles, marine snow and other particles, as this submersible came up to the surface. So we're looking out basically through the porthole of the submersible. And this is a krill right there. And watch this animal. The submersible's moving up, the animal's intrigued by the lights, and it paces the submersible as it ascends. Here comes another one in from the corner. Zip, moves up diagonally across the field. And our animal right here keeps pace. Then it gets bored and swims away. Okay, and this is probably the same species of krill that I just showed you the large-scale distribution for. So we know that these animals do have the ability to move vertically in the water column. Now, why would they move vertically in the water column? What advantage would there be to moving relatively small distances, some 50, 100, 200 meters, or even much greater distances, up and down in the sea? The ocean is vertically stratified with respect to many properties. This is a plot of the light gradients in the ocean, actually the light field. But this is light at the surface, 0 is the sea surface. We move down here to 400 meters, and you see that light decreases very sharply with depth. This is because the seawater molecules absorb light, and the light is also scattered and reflected off plankton and other particles in the water column. The ocean is also stratified with respect to temperature, and a zooplankton that was living right at the surface would experience a temperature of about 16 degrees Celsius. That same animal swimming down to 200 meters in this particular profile would experience a temperature of about 10 degrees. Now for a cold-blooded animal, that's a very sharp temperature difference. Phytoplankton, the plant plankton that forms the primary food source for many of these zooplankton organisms, is also highly concentrated near the surface. This is phytoplankton fluorescence measured as an index of chlorophyll A, and we find that they're sharply concentrated near the surface where light is higher for photosynthesis, obviously, and they also depend on vertical nutrients. Then ocean currents also vary with depth in the ocean. At least one component of currents that we call the baroclinic currents do, where an animal living near the surface would typically experience much greater horizontal currents than an animal living down deeper. In addition, predation pressure is very different in different sectors of the water column. The risk due to capture by sight-hunting fish and other planktivores is very different depending on whether the zooplankton is living in near-surface waters or down deep. So the prospect for finding a suitable habitat varies greatly depending on the vertical location that this animal positions itself in the water column. I want to show you an example of a few animals and the vertical migrations that they exhibit. First, we're going to begin with this wonderful copepod, Matridia pacifica. It's 2.7 millimeters in total length, and it's a small aquatic crustacean that is perhaps one of the most abundant animals on Earth. Now, how big is 2.7 millimeters? If your intuition fails you there, if you pick out a dime is 18 millimeters in diameter. So a little over 6 of these animals would fit end to end across this dime. All right? They're rather small, but they're very important in the ocean. Now, one of the devices we use to study vertical migration of these small animals is a very large device. It's called the Multiple Opening Closing Net and Environmental Sensing System, or MOCNESS. And the device is illustrated up here as it would look in the water when we're flying it off the New Horizon or a different script ship. The sampling device consists of 2 stacked pairs of 10 nets each, plankton nets each, that are streaming out behind the sampling device. And here it's being recovered on board ship. Then each of these nets is electronically triggered so that we can close it at a predetermined depth and step the device up through the water column, sampling the zooplankton in vertically stratified layers. We also include a variety of different electronic devices to sense the characteristics of the water in which the animals are living. Now, what do we find when we deploy the MOCNESS right out here in the San Diego Trough to try to understand this copepod, Matridia pacifica? These are vertical distributions of abundance. The black panels are the nighttime distributions, and the light blue are the daytime. We range from this ocean surface right here down to 400 meters depth. And we see that at night, this is 1 o'clock in the morning, just after midnight, the animals are aggregated mainly up in the upper 50 to 75 meters of the ocean. And 12 hours later, 1 o'clock in the afternoon, they've migrated down to about 275 meters or so. This very small animal undergoing a tremendous migration. The next night, they're back up at the surface. And now we've changed the sampling interval from every 12 hours to every 6 hours, and we can catch the dynamics as they sink at 6 o'clock in the morning, sink and swim, 6 in the morning, 1 o'clock in the afternoon, 6 o'clock in the evening, and then bang, midnight the next night, they're back up at the surface. The next day, they're down again. So the amplitude of this migration is, it's roughly 250 meters, the distance from the mean nighttime depth to the mean daytime depth here, about 300 meters. So the animal's migrating about 250 meters one way. It's 2.7 millimeters in length. And this means that it's swimming more than 92,000 body lengths one way up, and the same amount of swimming effort down. For a 6-foot-tall human, to put this in terms that we can perhaps relate to, this is equivalent to running 105 miles. That would be 105 miles before you had your croissant and your coffee this morning. You go about your day's business, run another 105 miles before dinner tonight. Now, clearly, they do this for a reason. They do this for a reason. I'd like to show you an example of another kind of organism, planktonic organism, a very large amplitude migration into the ocean's interior. These animals are called salps. They're actually your evolutionary relatives. They're planktonic tunicates. They have a complex life history where this is the solitary sexual phase of the generation, solitary sexual generation. They bud off a little stolon here, and this is a whorl of salplets in the aggregate asexual generation. And each of these organisms is about 50 millimeters in height or so, about roughly 50 millimeters. Now, they're quite common in the California current, but I'm going to show you an example of a vertical migration from the Atlantic, where a colleague of mine studied the salp, Salpa aspera, using the Mach Ness system that I described a moment ago. And here the sampling went down as deep as 800 meters, and we see that most of this population was down about 800 meters in the daytime, the largest element of the population. And they rocketed up to the upper 100 meters at night. Okay? Now 800 meters, I'm not very good with the English system, but this is nearly 1 kilometer, and that would be about half a mile. Okay, so they were known to swim about half a mile. But my colleagues were in that same area about a week later. They were present in a submersible that dived down to 2,000 meters, way off scale here. And notice the sampling in this study stopped at 800 meters. The submersible was way down here and found daytime layers of salps that were probably the same species. So it's possible, in fact likely, that these animals were migrating down much deeper than was detected in the sampling program, and perhaps as migrating up and down as far as 2 kilometers, a mile and a half or so every day and every night. Now, back to the whys. Why do they migrate? This is an illustration of different stages in the life history of one species of euphalzid, of krill. This is a larval stage, the Caloptopus. These are early Phersilia, juveniles, and adults. So this is all the same species in different developmental stages that they pass through. In fact, there are more developmental stages, younger stages that aren't included on this slide. Now, a graduate student here, Alex de Robertis, reasoned as follows. He said that if vertical migration is a predator avoidance mechanism, and if the risk due to predation is different according to the size of the prey, then the prey items that show the largest risk to attack by predators should show the strongest manifestation of vertical migration behavior. Expressed a little differently is, think of yourself as a sight-hunting fish. Now, obviously, the adults in this population are much more visually conspicuous than the little larvae are, right? The encounter distance at which you could detect these organisms is vastly greater for the largest body-sized members of the population. So Alex reasoned that these are the part of the population that should show the greatest propensity to vertical migration if their purpose is to avoid site hunting predators, and these should show the lowest propensity to vertical migration. In order to address this problem, he teamed up with a scientist here at Scripps, Jules Jaffe, who invented a rather ungainly looking apparatus, but it's very clever indeed. He called it the OASIS. That's for Optical Acoustic Submersible Imaging System. Now the OASIS uses a sonar, but it's very unlike the sonars used several decades ago. Much more sophisticated. It's a multi-beam sonar. It has 16 transducers, and it enables us to localize the three-dimensional positions of individual zooplankton in the ocean. It also enables us to take pictures of selected individuals. This is a digital camera right down here. This is a strobe flash that we filter to the red part of the spectrum so as not to modify the animal's behavior. And so periodically, if an animal swims into the right part of the acoustic field of view, if it swims up through here, it triggers a red light flash. We grab a digital picture, and we know just the animal that has triggered the acoustic device, and we know what's called the acoustic target strength of this animal. And then the current meters we use to get the three-dimensional current velocities. Okay. So, and these are images of some of the animals that were caught, these rather stunned fish in the upper left-hand corner, caught underwater as they swam past the OASIS device. They're juvenile walleye pollock and herring. And then these are some of the euphalsids that were filmed in that environment. This is a reference specimen shot in the lab. Okay, so we use the pictures to identify organisms and the acoustic device to track the three-dimensional positions. Now, back to Alex de Robertis. In this particular study site, which is a fjord in British Columbia, Alex lowered the OASIS device up and down over here, and he did this in the daytime and found the vertical distributions of the animals were concentrated between about 80 and 120 meters in the day. He did the same thing, lowered the device, and found the nighttime distributions. They'd moved up into the water column, and they're rather dispersed here. But this part wasn't a surprise. Up till this point, this is what you could do with conventional sampling devices and conventional acoustic devices. What was new was Alex deployed the OASIS device right here at 40 meters, and he sat and waited. And what he did was to wait to intercept the animals as they began their migration upward past the device, and then in the early dawn as they began their migration back downward. And the idea was to test the hypothesis of size-dependent timing in vertical migration, that the larger animals are the ones at greatest risk due to visual predators and should show the greatest migration behavior. What he found was the following. This is a plot that shows three different size classes of krill. We'll call them small here, medium, and large. And it shows the time of arrival after sunset of krill in the surface water, or near surface water. Remember, the device is at 40 meters, and the animals are starting to swim up past it, right here, like so. The time, sunset was right here. This is now 20.9 would be nearly 9 o'clock at night. 21.9 would be nearly 10 o'clock at night. And this illustrates the timing of appearance of these three size classes, where we see that 50% of the small animals arrived at this time, 50% of the medium animals at this time, and 50% of the largest size animals arrived at this time. So there was not a uniform timing of appearance in surface waters, but rather the small ones came up first and the large ones came up last. In the subsequent dawn, here's sunrise over here, just the opposite was expressed, where now the large animals were the first ones to descend down past the OASIS system, followed by the medium-sized classed animals, and followed in turn by the small, who came down just a few minutes before sunrise. Now, this means that the largest individuals are the last to ascend after dusk, and they're the first to descend before dawn. This is consistent with the predator avoidance hypothesis and the notion that the large-bodied animals suffer the greatest risk due to predatory attack. Okay. Now, I'd like to talk about a different kind of zooplankton. This is a largely herbivorous or particle-grazing copepod, Calanus pacificus, and it's a species that was made famous by the late Michael Mullen, who had a splendid career studying this animal and other zooplankton here. Now, a close relative of Calanus pacificus, Calanus marshalli, lives in the Oregon upwelling zone. The Oregon upwelling zone is characterized by very high concentrations of phytoplankton in near-surface waters. And what we're looking at here, this is a cross-section from the near shore. The beach would be over here, and the offshore ocean is out here. And this is the seafloor right here on the continental shelf. So a cross-sectional view across the California Current close to shore. And we see that the green layer, which is the regions of highest chlorophyll concentration, the phytoplankton that this particle-feeding copepod is most interested in, is sharply concentrated in the relatively near-shore waters and right up near the surface. So you would think the optimal location for this animal to live, if it were selecting a vertical place in the water column to live, well, that seems the obvious place to be, right about there, doesn't it? Well, but there's a risk associated with living there. And this doesn't have to do with predation. This has to do with currents. Since it's an upwelling zone, the wind-driven coastal upwelling drives near-surface waters offshore, and this results in deeper water coming up from below to replace that transported surface water. Okay? So this right here, this region where the food concentration was optimal, happens to be a region where the transport probability of being advected into the offshore domain is very great. And in the offshore, the food concentrations drop down precipitously. So what to do? This is an example of a daily vertical migration or diel vertical migration that results in a retention mechanism close to shore. The animals migrate up toward the surface where they feed in these phytoplankton-rich waters, but then they migrate back down into the deeper water, which is transporting them back up close to the coast. Up to feed, down, and this results in retention in the near-shore habitat where food concentrations are most suitable. Okay. Now an example, one of my favorites actually. This concerns a carnivorous copepod, Parayuchida elongata. It's a predator, but it doesn't hunt in the same way that the plankton-feeding fish do. The planktivorous fish require light in order to detect and capture their prey. The carnivorous copepods are essentially blind hunters. But what they use, you see these little hairs coming off the first antennae. They have long, outstretched first antennae and a series of small hairs, or setae, that move actually in the three-dimensional network. And these are very sensitive to hydrodynamic disturbances, to water flow disturbances in the ocean. So they detect their prey using these mechanosensory hairs stretched out on the first antennae. Now, what is the migration behavior, vertical migration behavior of our friend here, Parayuchida? Again, these are the daytime profiles here in light blue, the nighttime in black. In the daytime, both the juveniles and the adults live in deeper water. They avoid the surface waters in the daytime. And at night, whew, they come right up toward the surface. Almost all the juveniles come up to the surface, and about half of the adult females come up to the surface. It turns out that the egg-bearing females stay down and they don't migrate, but that's another story. But most of the population shows what we've now seen as the rather predictable migration of down in the day and up at night. Okay. So why do they stay down at day? Why not simply spend your whole day and night up in the surface waters feeding on small copepods, which are their primary preferred prey. And the explanation seems to be the risk due to attack by these site-hunting fish. This is a three-spined stickleback, Gasterosteus aculeatus, which is a primary source of predation pressure in that particular study site. And here's the copepod Pareuchida elongata being pursued, if you will, by this fish. Okay, so this seems fairly intuitive. But actually, the most interesting part of the story is yet to come. Those of you with sharp eyes will note that there's something else going on. You see this little object here in the figure. That's actually the star of this show here. If we enlarge this, we see that the carnivorous copepod Pareuchida is in turn in pursuit of a small-bodied copepod called Pseudocalanus. So this is a carnivore, and this is a herbivore. And in fact, the Pseudocalanus are the preferred prey of this carnivorous copepod. And the question is, what should the Pseudocalanus do to avoid its predator, which is migrating down by day to avoid the predators on the Pareuchida? This is a Pseudocalanus eye view of the Pareuchida in hot pursuit. This is the nightmare of a Pseudocalanus. These formidable second maxillae outstretched, ready to grab it. And this is just the beginning of the first antenna with these sensory setae that I mentioned that detect the mechanosensory disturbances. Well, my colleague Jules Jaffe, again, his laboratory created an animation of what it would look like for this predatory copepod to be in pursuit of the small-bodied Pseudocalanus. And let's see if we can run this here. So picture this on the left. These are small Pseudocalanus copepods, and this is nighttime, so they're swimming, they're grazing, these young innocents, and moving along. And then comes Euchida and nabs them. We have another view of a prey capture. Note the Pareuchida folds its first antennae down, flips its tail up, accelerates, flips out its second maxilla, and engulfs the Pseudocalanus. Okay, so what is the solution for this Pseudocalanus being pursued by the carnivorous Pareuchida? Now this is the Pareuchida migration that we looked at just a moment ago. Down by day, up at night, down by day, up at night. The Pseudocalanus show a reverse migration. The out of phase migration, where they're up at the surface by day and down at night. So they're in surface waters grazing on phytoplankton at just the time of day when their chief predators are down out of the water column. It's a neat trick. Okay, I want to convince you, or to show you, that it's not only the oceanic animals, but some algae, roughly plants, single-celled algae called dinoflagellates also can show vertical migrations. This is the dinoflagellate, this is a Goniolax spinifera, and this is a Lingulodinium polyedrum, which commonly forms a red tide in our waters. In fact, this is a suspension of cells that I collected during a red tide a couple of years ago and just put it under a microscope. This is unconcentrated, so this is a natural density of cells. But these cells have two flagella, two little whip-like flagella that they use to swim through the water. And so if a cell is going to migrate, it obviously has to have swimming ability. This is to prove to you that they can, in fact, swim. Single-celled dinoflagellates, they're about 45 micrometers. Roughly 500 of these cells would fit across the width of your thumb. Okay, so they do have the ability to swim. Now, at times, through their vertical migration, they aggregate at the sea surface in such exceptional densities that they're visible from the sea surface. They form what we call red tides. And this is an image taken by my colleague Peter Franks of a red tide of Lingulodinium polyedrum, just the cell I showed you a moment ago. This is an aerial shot of surface aggregation so intense that they're visible from aircraft, and some of the fine-scale distribution is caused by internal waves, which is a subject for another day. But the point is that these cells are attracted to bright light. So in the middle of the day, they swim upward and aggregate very close to the surface, and then they swim downward at night. This is another reverse migration. But you might ask, why would a plant show this kind of migration behavior? And the answer is that they migrate up to the surface by day to photosynthesize, but they migrate down, not as far as the zooplankton do, but 10, 15, 20 meters. They migrate down because nutrient concentrations are higher in the ocean, deeper in the ocean. So they migrate down by night to soak up nutrients, migrate up to in the daytime to photosynthesize. It's a neat trick. I'd like to show you what I think of as the shortest amplitude migration in the ocean. Now our spatial scale is very much different. We have to shrink ourselves down to the scale of sand grains. And what you're looking at is a cross-section through a beach. These are individual sand grains here. This is an artist rendition thereof. And you see down here is a layer of these little green rectangular cells. These are diatoms, the diatom Hantia virgata. And they show a tidally synchronized migration, where in certain parts of the coast, this species doesn't occur on our coast, but in other localities, in the summer, when the wave height is low, when the tide comes in, the cells slither their way down in between the sand grains to safe haven here, just a few millimeters down below the sand surface. When the tide goes out in the daytime, they slither their way back up between the sand grains and perch themselves on the sand surface to photosynthesize. But all this is happening on the scale of several sand grains in thickness. Okay, now one of the modern devices that we use to study vertical migration is one that, again, the graduate student Alex de Robertis worked with. This is a completely autonomous vehicle that is programmable. It has an onboard computer, and Alex could program it with a simulated migration behavior of different species of zooplankton. The device uses the Cartesian diver principle, where it shrinks the trapped airspace and then increases the density and sinks, or drives a piston out in increasing the enclosed airspace, and gains buoyancy and rises. And it has a GPS antenna communication devices. And Alex can deploy it so that it sinks down, stays at, say, the daytime depth of a zooplankton, migrates up to a nighttime depth of a zooplankton, comes right up to the surface, finds out where it is using GPS satellites, calls us and sends us data, and sinks back down. And this will give us, for the first time, the opportunity to track vertically migrating zooplankton populations through the ocean. Now, in closing, one of the synthetic views of vertical migrations was published by my colleague Vinogradov, who developed the idea of overlapping ladders of vertical migration. And the notion here is that animals migrating up in very near surface waters up here, this is increasing depth, going from the sea surface down very down deep, 8,000 meters into the ocean, that the migrations of near surface dwelling animals overlap with those of somewhat deeper dwelling animals, and this process continues into sequentially deeper depths in the ocean. As animals migrate up, they feed in shallower waters usually and migrate back down, that there can be a net transport of organic matter and energy from near surface waters into the interior of the ocean. And conversely, some of that can be transferred back up to the near surface waters. So this migration behavior has consequences for the ocean's capacity to sequester carbon and other elements. Migration behavior is also of great consequence when we think about the variability of marine fish populations, being able to predict fish population variability, because most of these fishes depend on zooplankton at some phase in their life history as prey. And of course, the migration behavior is also important in understanding sound propagation in the sea. But most of all, it's a fascinating phenomenon. Now, I would like to ask a little something of all of you, and that's when you're driving home from work in the evening just before dusk, or if you arise early in the morning before dawn, cast your thoughts to sea, to the open ocean, and think about this enormous biomass of oceanic animals that's moving up and down or down and up with the daily rhythm of the sun. Thank you. The question has to do with the abundance of food on Baja California and how that might affect the migrations of animals. First of all, it will affect the motile, the actively swimming animals, the nekton, fishes, squids, and, well, marine mammals, and so on. It's not likely to affect the kinds of organisms that I'm talking about that don't have much ability to swim horizontally. But certainly, one of the primary reasons that the California gray whale undergoes this Pacific Basin-wide migration has to do with the availability of food, which is actually the principal concentrations of food are in the Bering Sea area. So I think that prey availability for large motile animals can be a primary driver of migrations. Interesting question. The question is, how does El Nino affect the vertical migration? Well, the study of Metridia pacifica that I showed you was done in 1992, which was a year of relatively mild migration, relatively mild El Nino, but it was an El Nino event. The evidence was that for some species that have a behaviorally fixed migration cycle, that the El Nino doesn't seem to modify it at all. And that was the case in Metridia pacifica. There are other species that have what we call facultative migrations, that depending on the conditions in the water column, predator abundance, the presence of different kinds of thermal stratification, that they might show a stronger tendency to migrate or a weaker tendency. And some of those species, Calanus pacificus would probably be among them, would tend to show a reduced migration amplitude during El Niños. But another effect of El Nino is that it tends to introduce a different kind of fauna. We tend to see more of the subtropical Baja California zooplankton that come up into this area. And in fact, when that occurs, we see more vertical migration because the animals that dwell off Baja California are typically stronger migrants than those that occur in this water. So it's not universally true, but it's often true. So in short, it's going to depend on the biology of the animals. The question is, if a large number of fish are moving northward because of the warm waters, will that affect migrations? In principle, I think it probably would. If there's more, if the fish are plankton-feeding fishes, they're likely to introduce stronger selection pressure to force the surviving populations to be the stronger migrants. The question is, how much life is there under a square kilometer, a square mile of ocean? I can't give you a numerical figure off the top of my head, but I can tell you that, that the biomass of animals tends to decrease exponentially with depth. So that most of the biomass of animals that we're speaking of is going to be up in these near-surface waters. And the deeper dwelling animals down here are very, very much rarer, several orders of magnitude rarer. If you summed up the biomass of animals that was moving up and down in the ocean on a daily basis, up and down on a daily basis, it would vastly exceed the biomass of any other animal migration that we know of on Earth.
I'm sure Mark would be glad to entertain questions afterwards here, but thank you again, Mark, for a wonderful talk.